everyone. Welcome to the Thoughtworks Technology Podcast. My name is Rebecca Parsons. I'm one of your recurring co-hosts, and I'm with my colleague, Ashok. Ashok, you want to introduce yourself?
Hello, everyone. I am Ashok, one of your regular co-hosts of this podcast, and I'm delighted to be joining and discussing a very, very interesting topic today.
And we are joined by two of our colleagues from Thoughtworks India, Pooja Arora and Justin Joes. And we are here to talk about using reinforcement learning for drug design. Welcome, Pooja. Welcome, Justin.
Thanks, Rebecca.
So this project came about as a result of a hackathon, if I understand. Can you tell me a little bit more about the hackathon?
Sure. So this hackathon came up right in the middle of the COVID-19 crisis in 2020, where the world was fighting COVID and the scientific world was figuring out ways how to control COVID. That is the time where Government of India launched the Drug Discovery Hackathon. And as a part of it, there were three tracks to it. One was fighting a few of the COVID aspects. The another track was to build tools and algorithms that will help in the current COVID-19 crisis, but also thinking from a future pandemic perspective. And the another track was for any other moonshots. So we participated majorly in track 2 to build tools and algorithms frameworks for fighting such pandemics.
I think my understanding of this was like an open sort of like competition for many people to sort of explore different or novel approaches to trying to solve this problem, right?
Yeah. Or maybe I can just quickly share on. So we participated in three problem areas. One was to generate antiviral molecules, which peptides, which is in itself a different world and very different to find naturally. Using generative methods. Second was, if you've heard of, toxicity, drug toxicity. So can we early on identify the toxicity of potential drug molecules? The third one, which is the major one that we're going to talk about here, is can we utilize reinforcement learning methods for identifying the drug pose? And this was the one that, so we got with all the three problems, we got into the final phase. So it was divided into multiple phases in the final interviews or selection process. However, this particular problem got through the phase 1 and we got a 1-year project in phase 2.
I think given that there's probably quite a few different things in there, if you focus maybe a little bit on the last one, you said like using reinforcement learning for drug pose. Like what is drug pose really?
Yeah. So maybe setting a few synonyms or acronyms here. Look at when COVID-19 came in, right? It was something different. People were not reacting to the regular medicines. There were tests conducted, there were x-rays done, and a lot of lab tests done, which took us to identify that this is not a usual fever, or this is not a usual, this is a different, this virus is attacking somewhere else. That's what you call as the target. And for the identified target, I need something that will either help me stop that virus from spreading. It will attach itself, it will make some impact so that it does not spread further. So that is where the drug come into a picture. Now, taking the biology out of it, take an example of a lock and key. So the lock here is the target and key here is the drug. So unless you have the key in the right position with the right touch points, right orientation, the lock will not open. That is the importance of the drug pose. the drug will come and bind the potential drug. I'll cautiously use potential drug unless it is really identified. Potential drug will bind to the target, given the both of them are the chemical entities at the bottom, can bind in multiple ways. But binding in the right pose with the right interactions will only make it biologically relevant, and it will have its desired impact. Does that make
Yes,
sense?
it does. Oh yeah, definitely it does. It does clarify like what you were trying to, at least what you're trying to solve for. Yeah.
And how did you go about solving it?
Go ahead, Justin.
So the problem required us to kind of come up with a mechanism, specifically in reinforcement learning. So this problem has been tried to be solved in machine learning approaches, where you have data, you use data, generate a model, try to predict where that drug would bind to the target. Now, the problem was presented to us as a reinforcement learning problem, and we have seen reinforcement learning being used in, let's say, robotics or gameplays mostly, and there the expected outcome is finding a path. So we have the agent would try to navigate and reach a destination. So for us, the binding pose where the drug would finally attach becomes the destination, and a randomized starting position for that drug becomes a starting pose. So we would want to train the reinforcement learning agent to navigate it or push that drug to that final pose. So that is the overall idea of using an RL or reinforcement learning in solving the problem. Now, how did we kind of visualize the problem? Now, visualization was a bit tricky. Now, this is a three-dimensional structure, chemical complex three-dimensional structure. The analogy of lock and key, yes, it fits to make it understandable, but can we model it into a reinforcement learning problem? So we had to look at other areas which we can quickly understand how to kind of formulate it as a reinforcement learning problem. So in that case, parking lot came as a good analogy for us, but it doesn't capture the entire complexity. It just kind of gives an idea how the drug and the target would work. So here the target is the parking lot. There is a free parking space where I would want to park my car. The car becomes the drug. And I would want to train an agent, RL reinforcement agent here, which can drive the car into that parking space. So kind of making that problem map to a more robotic or gameplay kind of an approach. So that was understanding the problem. Now, once we have understood the problem, the second aspect is identifying the major, what will contribute to identifying that position. So now we have to look at chemical structures, atoms. The proteins are chemical structures in general. They are molecules, large molecules. So atoms come into play. How do I represent an atom? The drug again is a molecule which attaches to the protein. So how do I capture the idea of two molecules interacting with each other? So these would then come into the representational idea. And then one aspect of reinforcement learning is defining a reward function. A reinforcement agent kind of learns an action by repeating it, and the reward tells the agent whether the action is good or bad. And so designing that reward function becomes the third problem, or the third challenge in entire formulation of the problem.
I think I find the like you mentioned like the parking lot as an analogy in there. Clearly, I think the in the sort of key and the lock and key, or even like the parking lot, part of it was almost like it describe like the initial part of the problem they're trying to solve, which is actually where do you sort of fit in, or like finding the space. But then I suppose maybe a thing around, or do you like park the car facing forward of, or reverse in, that is the kind of analogy you're
Yes,
trying
that is the
towards this.
kind of analogy now. So you would want to park the car in the right orientation. You just can't just park the car. It has to be right orientation. You don't want to. Parking lot analogy kind of slightly makes the problem less complex. There are many complications which comes in when you look at it from a chemical space. You have molecules interacting with each other. So some atoms cannot be next to another atom because of repulsion. Some atoms would just attract and be there. So such complications come into play. Parking lot analogy was to kind of help us formulate the problem in a way which we can understand, we can relate to, so that we can kind of find a path from robotics, gameplay, and then finally apply it to a chemical space.
Yeah, it sort of reminds me of sometimes when you go into parking lot and you park a car, right, and you realize after you parked it, there's a pillar right next to it. So while you can park the car, you can't really get out. So it it's not really very helpful. So I can sort of see where that gets to. Okay.
Yeah, I was just going to say I will multifold that. When you have structures that are, they may look similar as a sequence of letters. That, there is a after that a G comes, at some point a L comes, but when that defines its final conformation as a 3D structure, those similar looking may give you a very different binding pocket is where we go, actually the drug goes in bind. Then ultimately the interactions that they form, the structure of the drug itself, so they give very unique setup. So you may get similar looking parking lots, but it's a tough job to get similar looking binding cavities or the binding pockets. So that adds another level of complexity, that the diversity that the agent needs to look at and to learn from is quite a lot.
And is there a simple explanation to the reward function on how you conceptualized that?
So the reward function here should help the agent kind of navigate this complexity while being able to capture the ideas around, that what atom should be next to it, what interactions should be there. So the reward function should enable the agent to find a pose which gives precedence to all of these. So to start off with training, you would take some data which exists. Okay, in this case, it is complexes which are similar for which we would want to train the agent to. In this case, SARS being the target for which we want to train. So we take similar complexes. Now these complexes, they have an experimentally attached drug, which experimentally has been proven that it works. And we would want our agent to mimic at least that to begin with. And once we are sure that the agent is capable of mimicking it, so because of the similarity between the SAS and this particular training complex, it can find the optimal pose for whatever complex we give. So with this as the premise, what we would want to do, the agent, so when we design the reward function, what we would want to focus on is how do I tell the agent that where you have reached and where you are, the action which you have taken to reach there, is it a good action or a bad action? And for this, what we take is a difference between where it has to reach. So it's practically a positional difference. So the drug would have to reach at coordinates. For simplicity's sake, let's take coordinates as an example. So drug has to reach at a coordinate of 10,9. That is the experimentally available position. The current action which the agent has taken has pushed the drug to 8,7. So a basic Euclidean distance between these two positions would give me a sense whether is the action good or bad. Has it moved it closer or has it pushed it further away from there? So if I use it as an absolute value, the changes between these individual actions would be really small. Now for the RL agent, I would want to tell that once you reach very close, I would want you to kind of push it further closer. But because once it has reached close enough, the changes in that distance is very small. The agent would be like, I'm not getting enough reward to do that. So I would want to shape my reward in such a way that the closer it gets a higher reward versus when it is very far off. So that would come under reward shaping in the reinforcement learning problem.
Almost like, when children play, they're trying to find something and then play hot or cold, or you say sort of freezing as to how far away you are.
Yes.
Similar sort of concept analogy in there, right?
Yes.
Yeah. Excellent analogy, Ashok.
Okay.
We always talk in terms of parenting. That's how
exactly.
That's how the reward function works.
Yeah, I can sort of see that in this case. So So it's almost like you like so does that mean like where you sort of described just following on from what Rebecca was asking around like the reward function, like this basic concept, is that the same, or does it like would that basic concept apply across different types of like these drug poses, or would you have to like tailor the reward function? So say you look at like SARS COVID as a thing, as if you're looking at a different type of problem, would you also have to tweak like reward functions and its principles, or the principles remain the same?
Okay, so we have to look at the current problem in two separate lenses. So the first one is, do I want to generate a model which can be then reused across multiple drug complexes, or do I want to do a one-step optimization? So one-step optimization would be across multiple iterations, the same drug and the protein complex are kind of played around, and they've eventually converged to an optimal position. So that would be one-step optimization. Reinforcement learning can be used there. In that case, I can use a tailor-made reward function, which very specifically can work for that particular complex, drug complex. But on the other hand, if I'm trying to generate a model, which after being trained on a large data set can be applied back to any complex, then I can't use that tailor-made individual complex-based tailor-made reward. I'll have to use a single reward, which in a way applies to all of the drug complexes which we are working with. So in this case, the distance-based is the most general way to look at it. So what I'm telling the agent is, all you have to do is reach to the target, and I'll tell you, have you reached the target? And while you reach there, learn why the action which you took was good. So that would come under the neural net or the machine learning part of it. So underneath it, we have a neural net which takes in a state of features. So the input drug and the protein or the complex will have certain features which would be dependent on the molecule, the atom, the interactions between them, the number of edges, etc. And this entire input representation would be converted into a action space, which would say, given this, what action should I take? Okay, so this neural net would be trained based on the reward, and this neural net would be responsible for capturing those the relationship between features, interactions, which atom should come next to which residue or atom for that matter. So since we were trying to develop a model, we had to go with a single reward function which would apply across a collection of complexes.
Yeah. Also, if I can add there, it would also, so it may not be the only reward function thing, but we look at it underlying the exposure that the agent needs to have, the differences in the complexes in terms of their structure, interactions, and many more. So, what kind of exposure that the agent needs to have? At some point, we were in a position that there were too many variables for our model to learn on. There's a changing reward, there are features, the data is too diverse. So at that point, we said, okay, let's look at it in a fashion that we reduce the diversity for the agent. We give it some kind of similar structures, and that will also help us understand its learning patterns. Identifying that right data set, and which will give it enough exposures, which will give it enough opportunities to experiment and learn on the type of interactions or the structural aspects will eventually help it get to the right pose. And eventually the goal was to have probably a very difficult task in a biological world was to have a generalized model. As much as we can, we wanted to move towards a generalized model that could cater to multiple protein or target families. When I say families, they're literally in terms of, think of it as different viruses, if for an example. So a type of virus may belong to a family and they will have their own structural dynamics. Another type of virus will belong to another family. So we want to have a generalized model that might be able to cater to a large extent to multiple families, and then eventually there could be further training onto it. So that was the larger goal.
And what were the results? I mean, I know you've published a paper on this, but how well did it do?
Yeah, sure. So, like I shared that, we said that, okay, there is a lot of diversity. Let's reduce the diversity for the model. Understand what reinforcement learning can achieve, what learning capabilities the agent has. So we reduced our data set to looking at only SARS-1, a particular type of protein target, M protease, collected some data around it, and then we started looking at then we trained our model. And so the final results that we got, which we have also published, was to that so as a reward function we used, like Justin shared, RMSE, root mean square error, to identify how good a pose is to the experimentally available pose. So our model was able to reach to an experimentally available pose starting from something like 6.5 angstrom. Angstrom is the distance in which the distance is measured, to 3.2 angstrom, which was fair. Then we also looked at that, okay, it has reached here, but what kind of interactions is it forming? Is it, connecting any hydrogen to any oxygen, or is it making the right connections? So there are experimentally available tools. One of them is ligplot, which tells you, the actual interacting amino acids or residues. So we compared with that, and we observed that our model had quite good, had similar interacting residues to a great extent, which gives a lot of promise in its biological relevance. That if it is interacting with the right residues, it may be close to the biological relevance, which we have not tested in the experimental lab, but it gives us that promise. Also, there is a series of work that we did after that, which we are yet to publish. But we have been able to improve our model performance to go to an extent of starting from 0.5 angstrom, I mean, as close as 0.5 angstrom to 4 angstrom. So that's the journey that our model has covered. But yeah, the published results that are available are till somewhere close to 3 angstroms.
That's right. I think some of the things that you were mentioning in terms of this, like clearly where you started off with, you sort of trained the RL models to, again, some training data sets which had some outcomes which were experimentally sort of in terms of like how do you sort of see this taken forward? So like the way these models would then be potentially used, is that about like reducing the the universe of options available so that then it can be targeted to try out a fewer set of in a more limited set of options that can then be tried out experimentally? Is that the direction or direction of travel for this?
Yeah. So eventually, as a larger goal, it should be able to help a structural biologist to take a set of a drug and a target pair and say that, I have this pocket, I have these couple of drug molecules which I feel are could be, a good potential for these, and I need to know the optimized pose for them. So there are a lot of methods available today. Some of them are traditional, some of them are deep learning methods. After that as well, there is a good amount of work that a structural biologist will do or an experimentalist would do. So a traditional method, some of the traditional method, what they would give them is how good a binding is. They calculate by a term is binding energy, by calculating the binding free energy. Hey, here are the 10 poses which are good, ranked 1 to 10. Now you can choose from them. So that is the outcome of that tool. So now the experimentalist will use a lot of their acquired knowledge to identify, okay, even the binding energy is less, this pose is not right. maybe because it doesn't form the right bonds. They can clearly see a structural hindrance that is there available, and many more. We believe that reinforcement learning could be path breaking there, where one, it will give you one optimized pose. With my learning, this is the optimized pose for you, and eventually it should be able to that's the larger goal, that eventually it is able to acquire that knowledge that is either available publicly or available in the way experimentalist interacts with tools, or acquire or utilizes their knowledge. So the agent being able to learn those nuances of scientific domains and being able to apply them to predict or to optimize, to get the right optimized pose. And this is one of the aspect. There are many more aspects in the entire drug discovery cycle where these could be used. And so to answer your question in very short, we hope that utilizing this model, one, it will reduce their overhead to look at 10 different things, utilize, and then come up with one. Two, it will also help them to try out for the unknown targets, the unknown drug target pair. So this is a lot of training has been done on the known drug target pair. Eventually, when there is a new virus or a new microbe or any other target is available, for which we need to identify, they should be able to, based on the knowledge that it has acquired, they should be able to apply that, and the agent being able to learn that this is the right pose and this is the right drug that fits in.
And adding on, because we are using reinforcement learning here, we can also introduce the idea of live learning. So, going ahead, if there is, the agent makes a prediction for unknown kind of boundary-based complex target, which is not part of its data set, and based on the outcome, it can be reintroduced into the learned model. And also, since the reward function can now be kind of tweaked. Right now, we are just using distance as the reward function. And that is where Pooja mentioned that the knowledge aspect of it can be introduced along with the distance. So now I can tell the agent, as per my scientific knowledge, you have done good. Rather than just saying based on the experimental data, you have done good, I can say based on the experiment data plus my scientific knowledge, you have done good. So that is how you can now kind of redesign the RL, the reward function in the reinforcement learning itself.
And are you continuing work on this model, or are you branching out into other things?
Yeah. So like I said, we have a working model. We want to package it and give it to a couple of scientists in our vicinity and known scientists to test it out, see how it performs. And so we have a set of things that we want to eventually do, but based on what they test, how did they feel, did it help, how much of help it was, getting that feedback from them, and then getting back to it is the goal right now.
Pooja, Justin, what you've sort of described is like quite fascinating. I think it opens up lots of possibilities, and clearly how technology can be used to accelerate drug discovery. But now, if you think about actually how to try and get this to, like, can you talk a little bit about the kind of technology stack that he used, and the amount of effort that actually goes into like training the model, and like the kind of data sets that are maybe available today, and do we need to get like better at collecting them as well?
So initial choice of the initial first phase, that would be when we presented it to the committee. Before that, we started with a 3D CNN approach, where since the drug and the target are three-dimensional structures, it made complete sense to just put them into a cube surrounding them as a three-dimensional entity. But soon we hit the performance problem, because 3D CNN, the cube is a densely packed information, where actually the drug and the molecule data is very sparse. It is, if I take a 10 by 10 Armstrong, 10 by 10 cube Armstrong, it would hardly have 200 dots inside them, which needs to be captured. So we had to then think of what is a better way to represent it. And graph CNN, graph convolution networks felt like a right approach there. But the only downside to it is we lose the three-dimensional information in graph convolution network. It's a two-dimensional representation now. So the challenge here comes like, how do I capture the idea of an atom being in space in a three-dimensional coordinate and another atom being its neighbor? So that neighborhood information is something which we need to capture now. So the three-dimensional idea can be captured as the coordinate itself, as one of the node features. So node here being the atom of the molecule. So three-dimensional coordinate captures the spatial information. Now the neighborhood information. Now, if we look at how a drug attaches to the complex, it comes pretty close, and then there are these chemical interactions which happen, and we would want the agent to know that such an interaction is happening. If you look at 3D CNN, it is purely proximity. The agent can understand proximity there, and it can deduce that, okay, fine, these two atoms are close, hence I'm getting a reward for bringing them close. Now, when it comes to graph, I have two separate molecule structures. One is for the protein, which is an independent graph. Another is the drug or the ligand, which is another independent graph. These two are not connected in any way, just purely from the chemical structure perspective. These have weak interactions between them. So the spatial proximity is now captured as graph edges which connect these or represent these interactions. So whenever two atoms, one from a ligand and a protein, come close enough so that certain rules are satisfied, which says that, okay, fine, these atoms are close, now they can form an interaction. They form an interaction because of a rule. We introduce an edge, and that enables the agent to understand that these two atoms are next to each other. And also we are using message passing. So it's a convolution network. CNN, it would create grids, and it would convolute the entire information across multiple grids, create a representation in one grid. Similarly, in graph CNN, what it would do is it would take the information of one node, convolute it with the neighbor, and based on the number of layers we add, it can accumulate, or one node can represent the information of n neighbors, n hop neighbors. So hop is the number. So in this way, the spatial proximity idea also gets captured as edges being added to the graph. And what graph would generate is a representation on which now the RL agent can take a decision. And the entire thing would be trained while the RL agent is training based on the reward. It tells, so there are two parts to it. One is it has to correct its action, which helps it do better in optimization. Second is it has to correct its understanding of the representation itself. So based on the reward, it has to do two things now. And it would be exactly the same when we do RL with any machine learning approaches. So it has to learn the neural net representation output of the neural net, and using that output, what action is best suited for that. So for this, we used DQN. DQN takes a state. DQN would be deep Q networks, Q-based learning network based on neural nets. It takes a state, generates intermediate representation within the neural net, and the output of the neural net is the Q values or quality or goodness values across multiple actions. So for us, the actions were delta translations in three-dimensional space. So I would tell the agent, move the ligand by delta X upwards, downwards, left, right, like that in 3D. So for a given input molecule representation, the intermediate network would churn out the value which says which action would be the best suited. And this would be reinforced using the reward function.
So a combination of graph-based as well as DQN. Okay, cool. Thank you. So
one major challenge was graph in DQN. Back then when we started, the literature was very sparse on this. There were, graph CNNs back then were used mostly for classification tasks. They were rarely used in combination with DQN. So that was one of the challenges. So trying to build was one challenge. And the second one, again, RL in drug also was one of the challenge. There was very little literature around it.
Yeah, there's hardly any literature in RL. So there are, like us, there are a couple of papers which say, which expand the potential, but very smaller experiments are available. So there were a lot of things that we had to experiment from scratch, and then do a lot of test. One of the example, if just you want to share on the where we were stuck on, there is some problem, but we didn't know what it is. We broke down the problem in terms of data, in terms of graph, and then we did some experiments to see that is our graph working right? Is it able to do a basic classification also?
So the representation, converting the representation to the right learning in the graph, and then then attaching the reinforcement learning part to it. So first we formulated it as purely a classification problem. So we would randomly spawn the drug and on left, right of the protein, and we would ask the classification to classify it as left or right. And by tuning it, we understood that fine, we have a graph model which learns now. Now we can use it with the DQN approach to do the reinforcement learning part. And the same went for the reward functions also. So, coming to RMSE, it might sound simple, like one step, we take the distance, we minimize it. It's a good idea. But identifying the right, the way you would shape the reward was a very difficult task to start with. So how do I tell the agent that you're pretty close, how you have to go further closer? So reward shaping in itself it's a complex space in reinforcement learning.
It sounds like there are quite a few different areas where you've had to both either come up with new novel techniques to try and actually solve for the end goal, as well as both in terms of like solving the problem around drug discovery, but also in terms of the building blocks that are necessary, both around RLs and getting graph CNs going. So yeah, well done. I think for people who would be interested in diving a lot deeper into this, I think you had mentioned the paper. So is the paper is published at the moment, right? Okay, great. Yeah.
Well, I think everybody understands the critical nature of the problem that you're trying to solve. We're hearing in the news about a new strand of bird flu that is going through the population, and we know that sometimes those things do jump to humans as well. So anything that can help in identifying a good target and drug pair is clearly critical. So congratulations on the success that you've had so far, and thank you very much for talking us through this and putting it in terms that mere mortals can understand. So thank you, Pooja. Thank you, Justin. And thank you, Ashok. And thanks, everybody, for joining us on this edition of the ThoughtWorks Technology Podcast.
Thanks, Rebecca and Ashok. Thanks for having us.